Hello, welcome back to New Scientist Weekly. This is your curated selection of the week's science stories. I'm Rowan Hooper.
And I'm Clare Wilson. Welcome to the show. It's episode 188, and we're recording this on May the 3rd. Coming up this week, we hear from aliens, or at least we hear when we might theoretically hear from aliens, and we meet the Lifeform with 7 genomes.
We're also going to be looking at what COVID does to your gut microbiome, and we're going to be hearing this.
Was that your gut microbiome, Rowan?
Oh, God.
I hope you haven't just stopped everyone from listening.
That's almost stopped me listening at that. No, don't be put off by grunting. There's more of that to come, but you'll find out why. Joining us on the pod this week is news editor Alexandra Thompson and reporter Michael LePage. We're going to start this week with an amazing story that you've been reporting on, Claire, about near-death experiences.
I love the music.
Let's start with a recap of what are they? What are near-death experiences?
These are those strange experiences reported by people who came very close to death. So typically their heart would have stopped beating and they only survived because they got medical attention and their heart was restarted. Now, sometimes while people were apparently unconscious, they report having some bizarre visions. These include the sensation of moving down a tunnel towards a bright light, the sensation of seeing or hearing from deceased loved ones, or having an out-of-body experience where you kind of float up and see your own body from the outside. Sometimes even the experience of being in a higher presence.
Yeah, they classify the near-death experiences in a number of different ways. And as you say, some of them are classified as, a religious encounter, a religious experience. It is all fascinating stuff. But what's new about it this week?
Okay, well, a lot of people are very interested in near-death experiences. And some of those people happen to be doctors who actually work in resuscitation. So, the people who are actually putting the paddles onto people's chests.
Right, to resurrect
to restart
them, to bring them back.
To restart their hearts, yeah.
Wow, so some of the patients being resuscitated, they might be literally going down a tunnel at the time and then we drag them back.
Yeah, we don't know how common it is, of course. So anyway, some doctors who work at the University of Michigan have collaborated with neuroscientists to find cases where people happen to have sensors placed on their head to record their brain's electrical activity at the time of their death.
Wow, so these people they just happened to be wired up when they died. So
Yes.
How did they die?
Yeah, well, these were all people who were in intensive care, either after a heart attack or a stroke, and they were on life support, and the doctors had judged that sadly they had no hope of recovery and would probably die soon. And as normally happens in these cases, in consultation with their relatives, they decided to switch the life support off. And the brain monitoring happened to stay in place. So these were the ideal people to investigate this subject.
Do you remember the movie Flatliners?
Yeah.
This is what they did. It's medical students who deliberately knocked themselves out and then were revived by their comrades, and then they reported on what they'd seen on the other side.
Well, yes. Yeah.
But anyway, what happened? What did we find out in real life?
So they found 4 people who this had happened to, and in 2 of them, they found unexpectedly a surge of brain activity of a particular type that has previously been proposed as a sign of consciousness. This is very high-frequency brainwaves in specific regions of the brain, at one on each side of the head, kind of roughly where your ears are.
Is this supposed to be the signature of the near-death experience then, this sort of burst of activity?
Yeah, well, I mean, we cannot be so definitive about it. And I should point out that in these 2 cases, they don't know if these people even had a near-death experience. Because remember, all 4 of these people did die. So they didn't come back and tell us if they did experience anything.
Yeah, okay. So obviously we don't know unless we do the plot of Flatliners.
And it's obviously a really hard question to study unless you were crazy enough to kind of do it deliberately like they did in that movie. I think we could say the researchers have found possible signs of consciousness at a time when we would have assumed that the brain was incapable of any meaningful activity. So it is one small piece of evidence suggesting that some of these near-death experiences could have been subjectively real to the person. It doesn't mean that they really saw their dead relatives, of course, or God. Now that near-death experience story could almost have been worthy of the sci-fi alert, but we've got something even more sci-fi for you, I think. This is when we report on something that has already been written about in science fiction. Rowan, what have you got for us?
So this is about the search for extraterrestrial intelligence. And you know how TV and radio transmissions leak out from Earth and go on off into space?
Yeah, so you mean when they talk about how soap operas from the 1980s have already reached the stars?
Yeah, exactly. So a couple of astronomers have looked more carefully at what this means, but not about TV and radio, but about signals that NASA have used in its deep space network to track spacecraft that have gone right out into interstellar space.
Do you mean like the Voyager spacecraft?
Yes, so those are two of them. Voyager 1 and 2 launched in 1977. They're now they're out of the heliosphere, out into interstellar space, but they're still transmitting and receiving messages from us. And NASA uses these powerful radio antenna to send signals to them. And these astronomers now have sort of extrapolated from that to work out which stars these signals may have reached and when the earliest response could be received back on Earth if someone is there to receive them.
Making the assumption that there are aliens on planets around those stars who are able to interpret and respond to the signals.
Yeah, I mean, that's just a small assumption. But if you do assume that, these two find in this paper that the aliens, the reply, might reach us by 2029.
Oh, that's really soon. That's just 6 years away.
Yeah. I mean, I'm not going to put it in the diary and get ready for a phone call, but, they've looked at Voyager 1 and 2, which, as I say, have already left the solar system, but the other spacecraft are Pioneer 10 and 11 and New Horizons, which went past Pluto a few years ago on its way out. And they've mapped the signals sent to these spacecraft. They've used this catalogue of stars called the Gaia catalogue and found which star systems the signals would reach and when. And they found 4 stars that have already received these signals. We don't know there's planets there, but if there are, then theoretically, these signals could come back in 2029. And then the ones that are, the other signals have reached would be 2031 and 2033.
In theory. I mean, that theoretically is doing a lot of work there, isn't it? What stars are these?
They're not stars that are famous or, ones with exoplanets. We don't know really anything about them because they're very faint. But, certainly we don't know if there's planets, let alone if they're hospitable to life. So, yeah.
Well, they're already making a lot of assumptions. So why don't we just also assume that the stars will have planets?
Yeah.
Statistically, planets do appear to be relatively common. So it's probably a safe bet.
I think that is a safe bet, certainly safer than the assumption that, we're going to get a signal back, a phone call in 2029. Right, it's time for a break and some messages. We've got a couple of new weekend trips to tell you about from New Scientist Discovery. The first is the science of rewilding at Coombs Head in Devon, England.
That's from the 1st to the 3rd of September this year. Discover how nature is making a comeback in the scenic Devon countryside. Go to newscientist.com/rewilding to find out more.
And the other weekender is Mysteries of the Universe in Cheshire. And that's the 29th of September to the 1st of October. Spend a weekend with some of the brightest minds in science as you explore the mysteries of the universe. And that includes a trip to Jodrell Bank to see the iconic Lovell telescope. Go to newscientist.com/universe weekend for more on that. We're back. And Michael, you've written about this extraordinary organism that has no fewer than 7 different genomes inside it. What is going on here?
Yeah, so this story starts around 50 years ago when a naturalist in Germany collected an alga. It's a single-celled alga of a kind called a cryptomonad, and it's been kept growing in this collection in Germany ever since. And back in 1988, some researchers took a look at it under a microscope and they spotted what seemed to be bacteria inside these single cells. They wrote a paper about it, which has just been sort of sitting around until it was recently read by Emma George at the Scripps Institution in California. And so her team asked for a sample of this alga, and they've now sequenced it and found out exactly what's inside it. And what they've discovered, not only does this alga have a bacteria inside it, it's actually got two different kinds of endosymbiotic bacteria. And one of these bacteria is infected with a phage virus.
Okay, right. Okay, so I've been keeping count here. We've got the
Yeah.
we've got the algal cell, and then two bacteria plus a virus, three distinct genomes inside. So how are we getting up to 7?
Okay, so to explain that, we've got to go back 3 billion years, and that's when one bacterium started living inside another simple cell and formed the first complex cell. So that bacterium became the mitochondrion, the energy-producing structures now found in all the cells in our body. And the mitochondrion still has its own genome, which means that all complex cells have at least 2 different genomes. So we've got the main one in the nucleus of the cell, plus the mitochondrial genome.
So when we have this process, one cell living inside another, that's known as endosymbiosis.
Yes, and around 1 billion years ago, it happened again when one complex cell acquired a photosynthetic bacterium, and that event gave rise to all plant cells, with the photosynthetic bacterium becoming a chloroplast, and the chloroplast has also kept its own genome, so all plant cells have got 3 different genomes.
Okay, so the 3 genomes are the different kinds of genome, right? So because there's lots of copies in each organism.
Yeah, we're not counting the copies, we're just counting the different types. There can be thousands of copies of the mitochondrial chloroplast genomes.
Okay, so plant cells have got these 3 distinct genomes, and this alga has these extra 2 endosymbiotic bacteria and the virus. So that's 6. Where's number 7?
So that is because even though this thing is called an alga, it's not actually a plant cell. So cryptomonad algae, they actually started out as these free-swimming predatory cells, and at some point they gained the ability to photosynthesize by engulfing not a bacterium, but an entire plant cell in the form of red alga.
Okay.
So this is where it gets really complicated. So the nucleus of that red algae has been retained alongside its chloroplast, which means that all cryptomonads have got 4 different genomes. They've got their main genome in their cell nucleus. They've got the shrunken remains of the red alga nucleus. They've got their mitochondrion and they've got their chloroplast. So then you add the extra 3 of the 2 endosymbiotic bacteria and the virus, and you've got 7 in total.
That's an absolutely extraordinary mixture here going on. What about the virus? So inside the bacteria, one of the bacteria has got a virus inside it, and that is being passed on when the cryptomonad host divides. So the virus is kind of not completely pathogenic then, like we expect viruses to be.
Yeah, we don't know for sure why it's not killing off the bacterium it's infecting inside these cells, but it actually looks from its genome as though it's actually helping the bacterium interact with the cryptomonad host. So it may actually be one of these rare symbiotic viruses that actually helps its host rather than hindering it.
And what about the other endosymbiotic bacteria?
That's not clear yet. Emma George is trying to find out, but she thinks that these bacteria have been inside these cryptomonads for at least 4,000 generations since the first sample was collected. So that suggests there must be some kind of interdependence going on.
So that must be a record, is it? 7 different genomes in one cell?
Yeah, that's definitely the record. The most that have been confirmed before is 6, but George was telling me that she's pretty certain we're going to find other cells that beat this record.
Wow. Okay, we've got some COVID news, which is something we've not actually said for a while on the pod. I mean, many people still think of COVID-19 as a respiratory infection, but its impact can extend well beyond our airways and into the gut. And Alexandra, you've been working on a special report about this.
Yes, I have. So a 2020 study found that up to 1 in 5 people with COVID-19 report having some gastrointestinal related symptoms, whether it be diarrhea, abdominal pain, loss of appetite, which I experienced, or nausea and vomiting.
And are those symptoms linked to an increased risk of getting a severe dose of COVID or even dying of it?
The symptoms haven't, however, disruption to the microbiome has. So a team at the Chinese University of Hong Kong has been analysing the stool samples of people with the virus from early on in the pandemic, and they found that disruption to the microbiome, which they measured by an increase in these so-called harmful bacteria and a depletion in helpful bacteria, is linked to more severe COVID-19 and potentially even death. So it sounds like we don't know exactly what's to blame here. Even years into the pandemic, why is it so hard to figure out how the virus can impact our gut? Because so many systems interact in our gut. You've got the walls of the gut producing enzymes that digest our food. The gut is home to trillions of microorganisms that make up our microbiome. Our gut influences our immune health. It even has connections to the brain, and all this interconnectivity makes it really hard to tease out what is causing what.
Well, what's the leading theory as to what's going on?
Probably the ACE2 receptor, which the coronavirus binds to when infecting cells. And this receptor is more common in the gut than the lungs, but there isn't strong evidence to suggest that the virus replicates in the gut. It could be that by binding to this receptor, there's disruption of nutrient uptake because ACE2 is involved in the uptake of the essential amino acid tryptophan.
And gut issues we know are or can be an ongoing issue with long COVID, can't they?
Yes, a 2021 study of people hospitalised for COVID-19 found that 16% of them had gastrointestinal complications more than 100 days after they were discharged.
And do we know why?
Well, one study found the virus's genetic material in the guts of people with long COVID, but the researchers couldn't grow the virus from the samples. So are they causing inflammation? Are they affecting the nerve endings somehow, affecting the microbiome, potentially impacting the permeability of the gut wall? We just don't know.
So that's not helping us develop any effective treatment with all these unknowns, is it?
Well, people have suggested probiotics and fecal transplants, but the evidence isn't really there to support these treatments. Clinical trials are ongoing, so hopefully we'll have results fairly soon.
Okay, thanks, Alexandra. And as I said, there's you've been working on a long special report on this for the magazine, and that's by science writer Michael Marshall, and we'll put a link to that in the show notes. Now, it's time... It's time for those lovely grunting noises and Lifeform of the week.
I'm not sure I even want to know what was making those sounds.
Well, it sounds very pig-like when you listen to it like that, but they're elephant seals, male elephant seals, alpha males, grunting at each other by way of assessment of each other's size prior to potentially, having a scrap.
Wow, they certainly sound like fine physical specimens. I'm sure if I were a female elephant seal, I wouldn't be able to control myself. I take it they are doing this to fight over females. And
yeah.
When you say that alpha males doing it, does that mean beta males don't do this grunting?
Well, as a male, I'll have to say all males grunt. It's what we do. So the beta males do it as well. But the alphas are, they do it in a deeper and stronger way, and they have to do it really to fight, to secure groups of females. In this paper that's just come out, they call them harems. That's what biologists call groups of females that are controlled by males. But it does strike me that we need a different name for this now, because harem doesn't seem quite right. But this week we're featuring them because this study shows that males with the largest harems end up dying younger than those with fewer females in them.
Yeah, so it kind of makes sense. I mean, are they expending a lot of energy or like trying to control the females? Or
yeah, I mean, the harems range in size from 5 to 50 females. So one male has to sort of try to fence off effectively this whole group of females, and you get subordinate males creeping along the beach. I'm just thinking of humans doing this, creeping along the beach, trying to mate with some of the females.
We've all seen it.
Yeah. Yeah, exactly. So the alpha male has to defend all of these. Rivals are constantly trying to, coming to challenge him. They're not called elephant seals for nothing. They're 5 tons, and so they have terrible fights. You often see them covered in blood, and they don't eat during the breeding season at all. So they're just lolling around on the beach, fighting and mating with females for months at a time. And one of the biologists who's been studying these is Kyle Lloyd at the University of Pretoria in South Africa. He noticed this weight loss and the loss of body condition and found that it was especially those males that had the most females. And then looking back through 34 years of records of the elephant seal population, he found that the males died younger when they had these bigger harems. So those males usually survived to 8 or 10 years old, whereas the females lived about 20 years.
But I guess it might be worth it as a strategy if they have more offspring. I suppose it's like live fast, die young.
Yes. Yeah. I mean, a successful male may father up to 200 pups in those 8 years of burning brightly. But yeah, then you burn out. But the subordinate males, surprisingly, those ones without a harem, they had even lower survival rates than the dominant males.
Oh, why is that?
Well, I asked Kyle this, and he said basically we don't really know. It could be genetics, as most seals are fathered by these alpha males, so that might be it. But there's a few other potential things it could be, so we need to look into it. You'll be pleased to know that's all the grunting seals for this week. Thanks to our guests Michael LePage and Alexandra Thompson, and thanks to you for listening. I'm Rowan Hooper.
And I'm Clare Wilson. Do subscribe to our show and tell your friends all about it. And we'll see you soon. Bye for now.
Bye.
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 This podcast is produced by OG Podcasts. Find out more at ogpodcasts.co.uk.